Hello and welcome to my presentation. My name is Antje Neubauer and I'm working as application specialist at the Enpresso GmbH, a small privately owned company based in Berlin. Today I'd like to show you how our EnBase technology works and how its Enpresso products simplify microbial protein production, while significantly increasing yield and reproducibility. If you are working with recombinant protein production in microbial systems, you are likely familiar with the common bottlenecks: low cell density, poor protein yield, and unpredictable results. Scaling up is difficult, and optimizing every single parameter from media to induction can be both time-consuming and frustrating. On top of that, conventional media don't mimic industrial fed batch conditions, which create serious problems during scale-up. Our solution is based on a simple but powerful principle: controlled glucose release for a healthy, balanced microbial diet. Conventional cultivation media often create an all-you-can-eat situation, or on the other hand, leave the cells starving, both of which stress the cells and reduce performance. With a balanced, steady nutrient supply, cells grow in a healthier, more productive state. This is solved by a fed batch strategy in industrial bioprocessing, where an external feed pump is used to add concentrated feed solutions in a controlled manner. While this approach cannot be easily applied to small-scale shaken cultures, we have developed an elegant solution. Instead of using an external feed, we supply a soluble sugar polymer to the culture that is not a growth substrate for the cells. Additionally, we add a biocatalyst that cleaves this polymer and releases sugar monomers which can then be consumed by the cells. By adding this enzyme in catalytic amounts, the system functions in exactly the same way as fed batch system with an external feeding pump. Initial tests with Escherichia coli in 96 well plate format confirmed that the idea really works. All wells were filled with the same medium, with a polymer instead of glucose, with the same amount of inoculum, but the amount of biocatalyst was varied from well to well. As I told you before, the polymer is not metabolized by the cells. In wells without biocatalyst, no growth is happening. This is the lower black curve. In wells where only little biocatalyst is added, we see a constant linear growth. In wells where more biocatalyst is added, the biomass increases higher. And this continues in this example up to a biocatalyst concentration of 3 units. However, in wells where the concentration of the biocatalyst is even higher, we see a decline, as in these cultures glucose accumulates and is not anymore controlling the growth. This has the consequence that the culture gets oxygen limited and side metabolites are formed, the products of the typical mixed acid fermentation of Escherichia coli. I want to highlight here that already in this early phase of development, ODs of 20 to 25 were obtained, which is much higher than what can normally reach in 96 well plates. Our product kits with ready-to-use growth systems are optimized for Escherichia coli cultures for different applications and for Comagatalia puffy. For customers from the industrial field, the most interesting product is EnPump 200. It contains the soluble polymer and the glucose-releasing enzyme and can be applied in every individual customized medium. Enpresso is designed for practical workflows. Simply dissolve the pre-optimized medium tablets in sterile water. Add antibiotics, the glucose-releasing reagent A, antioinoculum, then start shaking. By adding the biocatalyst separately, you have the opportunity to optimize the feed rate according to your needs. Otherwise, you only take the recommended concentration. After overnight cultivation, just induce the culture the next morning. You don't need to monitor UDI 600 for induction time. The protocol is aligned with a normal working day for convenient use. After 24 hours of expression, reaching a final OD 600 of about 20 to 30 in shake flasks, you can harvest the cells in the morning. Independent labs have reported dramatic improvements. For example, a 60-fold increase in toxic human lipoxygenase expression at Charité Berlin and a 36-fold increase in dehalogenase production. These aren't isolated results. Enpresso has been cited in over 200 peer-reviewed publications to date. Now I want to talk about some case studies done in Escherichia coli. Joseph Circus describes in 3 articles the successful optimized expression of the very difficult-to-X protein, the ribonuclease inhibitor. The gene was first cloned in the plasmid library with 45 vectors. To measure the protein aggregation, a second plasmid with luciferase as reporter was inserted. Expression conditions were analyzed by luciferase measurement, soluble protein quantification, and SDS page showing total soluble and insoluble protein. All of these analyses performed with cellular material derived from just one 96 microwell were possible due to higher cell densities achieved through cultivations using EnBase technology. The influence of the growth rate at the time of induction has been investigated in shake flask cultivations by induction at different time points. It's noteworthy that the best working conditions, namely to induce at a specific growth rate of 0.22, was verified by a fed batch cultivation in a bioreactor. In addition, it shows the importance of providing screening conditions in fed batch mode. As you can see in the graph on the right, cultivation in LB medium ended with a bad result. In the strain screening activities had been performed in LB with batch mode, this strain would never have been selected. This graph shows very nicely how well parallel cultivations can be performed with the EnBase technology, as an almost synchronous growth of 4 times 96 well cultures is observed before the temperature shift. You may know that there is a growing interest in the production of antibodies in prokaryotic systems. In this review, the authors from New England Biolabs show a long table of results on the functional yield of various antibodies. Studies with Enpresso products were successful and even led to better results when compared to conventional media. The third example shows the production of the peptide valinomycin. In the first step, the expression of the very large protein valinomycin synthetase with 2 subunits of greater than 100 kilodalton each must be successful. In order to be able to produce valinomycin subsequently, the culture must still be in a metabolic state. To obtain the valinomycin synthetase in soluble form, Yaitzik and colleagues tested different tags and ribosome binding sites, different E. coli strains and culture media, and user concentration and temperature. The use of Enpresso grow system had a major advantage here. The slow, controlled growth allowed cultivation for up to 100 hours, which not only increased the soluble form of valinomycin synthetase, but also expressed valinomycin in high concentrations. To further prolong the culture, they added the polymer at different points in time and achieved a final OD of more than 50 and even higher valinomycin yield after 50 hours of cultivation. Now I will talk about some applications in yeast. Yeast generally grows more slowly than Escherichia coli, so maximizing growth is particularly beneficial. In addition, in Vitrogen's standard protocol requires 1 or 2 pulses of methanol addition per day. The values shown in the graph illustrate the effect of pulse feeding with methanol. Because the cells also metabolize methanol as a carbon source, fluctuations in methanol concentration can cause stress. By using Enpresso with a constant background feed, carbon limitation is avoided. Using Enpresso Y-defined means less hands-on time. The tablets must be dissolved in sterile water. No media changes are required during the cultivation process. No OD measurements are required. You can grow your culture overnight and induce in the next morning. NPAM 200 allows exponential feeding. Last year, the Laboratory of Bioprocess Engineering at TU Berlin has published the mathematical model to better understand the Enpresso system and also to enable exponential feeding to keep the growth rate constant. The first of these models has just been published in Bioengineering 2024 by Kemmer et al. At the robot station, the Enpresso system was used, where the specific growth rate could be kept constant over a longer period of time by adding the biocatalyst or polymer. You will benefit from using Enpresso products if you need to make recombinant proteins in Escherichia coli or Comagatalia, as they support microbial growth. Higher final cell densities allow you to reduce your cultivation volume and so save time. It's very easy to use as the Enpresso growth systems are ready to use cultivation media. Reproducibility through controlled growth with Enpresso, even on a small scale, like a 96 well plate, provides you with a perfect environment for screening for expression conditions to develop a bioprocess on a large scale. Finally, our company is looking for teams who face challenges in high throughput expression of clones or simply need higher and more consistent yields in protein or enzyme production, including reproducible and scalable cultivation systems. You are welcome to contact us if you have difficulties in functional protein expression and aim to optimize and scale your experiments from high throughput screening to industrial production. You are the right partner if you seek to reduce variability in microbial processes and want to enhance your production efficiency. Thank you for your attention. You can reach out us via sales@enpresso.de and I'm happy to answer your questions now.